Okay, thank you. Hi, everyone. Thank you for joining us for another CNC meeting online. Thank you for your presence. We today welcome the invited speaker, Anke Konrad. She's a researcher at the C3C Centre with the talk entitled The Rate and Landscape of Accumulating Mutations in C. elegans, Genomic and Transcriptomic Considerations. Thank you, Anke, for accepting our invitation. Before I give the floor to researcher Ivo Cello, who will present Anke, I would like to invite everyone that is following this presentation to invite you to put your questions during the talk in the chat that you have on your right on the YouTube window. Sorry. So now I would invite Ivo Cello to please give you the floor and to say a few words and present Anke, please. Thank you.
Sure. Thank you, Marta. So it's a great pleasure to welcome Anke Konrad. She has come recently to our lab, and we're very excited about the work she's going to do. So in her CV, you can see that she moved early on for graduation and master's at in New Mexico State University in the United States. And then she did her PhD on evolution of gene families by gene duplication with David Liberlis, which I think some of you might know, at University of Wyoming. And then after her PhD, she did postdoctoral work with Aishali Kachu, University of Mexico, and then in Texas, AM. And where she was looking at the fitness and rates of C. elegans mutations, I think is the work she'll be talking mostly about today. And then later on, she did a short period with Isabel Gordo at the IGC, where she worked with mutations in the coli, evolving in the gut of mice. And now she's with us, and she's going to start this very exciting project on health and evolution with C. elegans. And well, I guess that's it. Without further ado, Anke, the floor is yours.
All right, thank you. I need to share my screen. Is everything good? Can we see it? Okay.
Yes.
So as Ivo implied, I will be talking about the evolution and the mutation accumulation in C. elegans. And before I go into the more data-heavy part of the talk, I would like to start by giving a quick review of why one might use mutation accumulation experiments. So what mutation accumulation experiments allow one to do is to propagate a population of an organism, in this case C. elegans, at the minimum population size, which allows you to diminish the effects of selection and hence get an idea of all of the mutations that arise and may accumulate other than those that are most lethal or sterile. And that in turn allows you to get an idea of what the spontaneous mutation rate in that organism is. So it essentially gives you the raw input of variation. And from that, or on that, selection may act, and that essentially gives you the bottom line of what evolution may occur. And of course, this is a very important aspect of adaptive potential and also genome integrity. So, well, a few reasons of why C. elegans is a perfect organism for mutation accumulation is because one, it is a multicellular organism, so it is a little more applicable to humans than E. coli might be. It is fairly easy to propagate in the lab. You have a generation time of 4 days. It is fairly easy to keep your generations non-overlapping. And the transparency of the worm allows for easy identification of life stages so that again, the propagation is fairly straightforward. So the mutation accumulation in these experiments of my last 5 years of my life were actually done before I joined the lab. So all of these experiments took 9 or more years if you include experimental and data analysis. But essentially, here the mutation accumulation was taken one step further in the sense that in addition to having a single population size at the minimum size possible, we also propagated these worms at 2 larger population sizes, so of size n equals 10 and n equals 100. In these larger population sizes, the idea was to essentially see how increasing efficacy of selection might affect the number and types of mutations that are accumulated over time. So these lines at size 110 and 100 were propagated for over 400 generations, which took about 4.5 years of hard work by Charlie Kachu and her undergrad students. And at the end of these experiments, where you had the 20 lines of size 1, 10 lines of size 10, and 5 lines of size 100 worms, as well as the ancestral worm of this experiment, they were all sequenced so that we could identify every mutation that had occurred throughout the evolutionary phase. Now, the rationale, which I hinted on, for this experiment is that it is expected that with increasing population size, the mutations that have very large negative effects in this case should be weeded out by selection. And these most deleterious mutations should only accumulate in the smallest population size, while those of medium effects might accumulate in the small population size and the medium population size, and then the mutations that have very low negative effects may accumulate in all 3 population sizes. And this rationale we tested in 3 different mutational classes. We looked at mitochondrial mutations, large gene duplications and deletions in the nuclear genome, and then small nuclear substitutions and insertions, deletions. And I will start by talking about the mitochondrial mutations. Now, the first thing to keep in mind about these mitochondrial genomes, or the mitochondrial genome dynamics, is that the inheritance of these genomes is not non-Mendelian. First of all, you have a haploid genome rather than a diploid one, and any given mitochondrion can have actually more than one genome at a time. But what this comes down to in the end is that if you have a cell with many mitochondria, you essentially have a population of mitochondrial genomes rather than one single genome, which might be the case for the nuclear genome. So heteroplasmy itself, meaning that you may have more than one mitochondrial genotype within a cell or organism, is an issue here. And since you have a population of mitochondrial genomes, every time you have a cell division or a new generation of the host, you essentially see a population bottleneck of the mitochondrial genome population. So if you start out with any given population of mitochondria and go through a number of different cell divisions, different tissues, different cell lines, they can all have a different makeup of the original population. And that is also important to keep in mind. And of course, these haploid genomes are inherited uniparentally, at least in animals, with very limited leakage and usually no recombination, which means that the variants within a mitochondrial genome are usually linked, and a deleterious variation may hitchhike with a beneficial one, or vice versa, whatever the case may be. So what it comes down to, or one way to look at this, is that mitochondria have their own effective population size within an organism, or in this case, within the germline. And then when it comes to actual fitness effect of a deleterious mutation in a mitochondrion, there are two ways to look at this. The first one is the deleterious effects on the host, which can be direct or indirect. In the direct case, it implies that the germ cell maturation or the offspring survival is affected, and that this variant is passed on to the next generation. The indirect effect of a mitochondrial mutation might be in a somatic line. It's not heritable because it might be tissue specific, but the mitochondrial variant can still affect the lifetime reproductive output of its host by causing disease or lower its survival. Now, just because a variant is deleterious to the host doesn't mean that it is deleterious to the mitochondrion itself, though. It could be beneficial to the mitochondrion because it might help the mitochondrion reproduce faster. So essentially what you're looking at then is a mitochondrial selfish element. And to some extent, the propagation of these might be tolerated by the host before they adversely affect the health of the host. So even though the variant is deleterious, it may not be purged effectively from the population. But let's take a look at this. So the first thing we looked at was the mutation rate of the mitochondria. And the way this is calculated is to essentially take all of the mutations in a population and add up their population frequency and divide that by the number of bases and the number of generations through which these mutation accumulation lines were propagated. This way, you avoid the, you essentially take into account that the mutations that arise have the likelihood of going to fixation that is equal to their current frequency in the population. And you avoid overestimating your mutation rate in these mitochondrial genomes. So when you look at the actual mutation rate by population size, even though you do see a negative trend, which is what you expect if negative selection is acting on the mitochondrial variants, you do get a negative trend, but statistically speaking, this is not significant. So you can't say that there definitely is selection, though it may be hinted at. However, if we compare mutations that are synonymous or intergenic and likely have no effect on the fitness to those that are in genes and are non-synonymous or potentially frame shift mutations if they are indels, then we do not see a trend with population size for the putatively benign mutations, but we do see a significant negative trend with population size for those mutations that likely do have a functional change associated with them. And hence it does look like for the non-synonymous and indel mutations, there might be negative selection that acts upon them in the larger population sizes. When we then compared our results to natural isolates, the kind of mutations that occur in natural isolates and have fixed in natural isolates. The spontaneous mutations are very heavy on the replacement substitutions and indel mutations, while those mutations that go to fixation in natural isolates are predominantly silent. So this implies that the very strong selection due to very large population sizes in nature largely weeds out the non-silent mutations and just retains silent ones. Again, this is due to strong selection. And the genome composition of the mitochondrial genome itself, which is very AT rich, seems to be a product of the mutational spectrum itself, which is primarily G to A and C to T transitions. And that is true across species from Drosophila, Daphnia, and the 3 different nematodes here, Pristioncus, C. brixi, and C. elegans. In all cases, the mutational spectrum very well explains the genome composition. So despite the fact that the mitochondria have bottlenecks and hence population sizes of approximately 60 mitochondria per generation, we estimated this. I didn't actually show the calculation, but it is part of the paper. The host effective population size still affects the mutation in mitochondria and the accumulation of the mutations in larger population sizes. Selection acts mainly against the deleterious variants. And even though deleterious variants are selected against, one thing that we did see was that mutations that mainly delete part of a gene still remain in the population at low frequency, which implies that there is an advantage for the mitochondria. But if the frequency rises too high, that's when they become deleterious for the host and are purged or kept at bay at the very least. So what about selection on gene duplications and deletions in the nuclear genome? So just a very quick review, there are a number of different mechanisms by which gene duplications and deletions occur. For smaller scale duplications, the main mechanisms are either unequal crossing over or slippage of the replication fork. Both of them can cause up to medium-sized duplications. And then, of course, you have the other mechanisms like aneuploidy or polyploidy that cause the duplications of entire chromosomes or genomes. Now, gene deletions, of course, if they delete genes that are singleton genes to begin with, have likely deleterious effects, but gene duplications are considered one of the ways to introduce new genetic material into the genome that may be functional and upon which adaptive evolution can act. Whether that is by optimizing gene dosage, partitioning the regulatory machinery to essentially allow for tissue-specific expression or spatial or temporal differences in expression, developmental differences. You can have the resolution of promiscuous protein functions, specialization of different kind of reactions. All of these potential mechanisms can help in adaptation. And the null model for gene duplicates is the view that a new copy might initially be selectively neutral. Now, this has been challenged over time, but this is still the null model. And when we look at our initial gene duplication rates, which are on the left in green here, for population size 1, 10, and 100, again, we do see a negative trend in gene duplication rate with population size, but this is not significant, so it's a stretch to call legitimate selection on this data set right here. In contrast, the gene deletions have a very strong correlation with population size, implying strong negative selection against gene deletion, especially singleton gene deletion. Now, when you look at these graphs right here, there are two outliers in both of these plots, both in the population size 1 for gene deletion and gene duplication rates. And these are due to one of the experimental lines experiencing massive gene duplication and deletion events. Essentially, the chromosome 1 and chromosome 5 of mutation accumulation line 1T experienced duplications that encompassed hundreds of thousands of genes. So the rate of duplication and deletion is, well, skyrocketed due to this. And much of our downstream analysis from here in the size 1 populations excluded this line in order to avoid very, very positive results just because of whatever this line had in terms of gene duplicates and deletions. So when we look at the length or the span of gene duplications and gene deletions, both of them, both of the spans were smaller in larger population sizes, implying that the larger duplications and the larger deletions on average had a higher deleterious fitness effect. And this was true whether we included or excluded line 1T. For the deletions, there are a number of gene deletions in the larger population sizes that aren't fixed. They only occur in some of the individuals. If we include them, they're also very large, then our correlation of population size to deletion span is no longer significant, which implies that these segregating deletions may in fact still be under selection. Now, whether they are purged ultimately or not, I can't tell, but it would be my guess. The next thing we did was we looked at the gene expression of the duplicated genes. And one thing that we noticed is that while the gene duplicates in line 1T on average had a gene expression that was slightly less than 2 times that of the singleton gene, for the remaining lines of size n equals 1, the expression of the 2 copies was higher than 2 times the expression of the singleton gene, implying that the expressional changes of a gene duplication is not simply just 2 times that what it was. So it's not additive. And when you actually look at the change in expression for these size 1 lines, as well as for the gene duplicates in size 10 and size 100, you do see a negative correlation with population size, which again implies that there might be selection against more extreme changes in expression, which really implies that larger changes in expression are selected against in larger population sizes. And similarly, I'm going to start on the right in this slide. With larger population size, you also have fewer genes duplicated, which had a very high expression prior to the mutation accumulation. So genes that ancestrally speaking have higher, are more highly expressed, are less likely to be retained upon duplication. And in terms of gene deletions and expression, this should not come as a surprise. Deletions are on average more likely to happen in genes that are either not at all or very lowly expressed. So highly expressed genes, when deleted, are most likely very, very deleterious. So on average, they are not deleted frequently. And in addition to the expressional differences between between population sizes, we also saw a non-random distribution of duplications and deletions across the chromosomes. On average, chromosome 5 had the most copy number changes, both deletions and duplications. And on average, most copy number changes occurred in the arms of the chromosome, the arms of the chromosome rather than the core or the tip. And one thing that needs to be mentioned here is that the core of the C. elegans chromosome is usually more gene rich. It is more strictly more packed, and it has less repetitive sequence. So the sequence complexity of the arm itself invites more unequal crossing over or duplication itself, or deletion for that matter too. So this is not unexpected, but it is nice to see nonetheless. And finally, this is in large part due to the fact that most duplications span more than one gene or more than two genes in most cases. The vast majority of genes that are duplicated are complete in all three population sizes. Even though you do have differences between the three different population sizes, because on average the length decreases for the size N10 and 100, and hence you have fewer complete genes there than you do in the smallest population size. And there are two side points to this story with the expression and the copy numbers. The first one is the ribosomal DNA copy number. While it does not change with population size, all three population sizes we noticed significantly increase relative to the ancestral copy number. And one thing that maybe one day I can follow up on is whether or not this has to do with genome integrity, because there are theories that ribosomal DNA copy number may correlate with keeping the genome, keeping the genome integrity up, but it would be nice to test this one day. There's one other thing, and this, if you're interested in transposable elements, this paper by, with Olva as first author, just came out in BMC Biology. This is another part that we were looking at. The transposable element expression and copy number in these C. elegans lines also significantly decreases with population size, which also implies that not just the gene duplicates of protein coding genes, but also the gene duplicates of transposable elements are under selection in these lines. And this paper is really, it's a pretty decent read. So anybody interested in transposable elements, I highly recommend it. So as a summary for the copy number changes, gene deletions are generally under much stronger selection than the duplications. The ancestral expression of the gene duplicated affects whether that duplicate might be retained. The intensity of the change of expression upon duplication affects whether a duplicate copy is retained. And the spatial distribution of gene duplications and deletions is non-random and may also affect whether duplications or deletions are retained. Now, at this point, we saw that both mitochondrial mutations and large nuclear mutations are likely under selection. So in this last part, looked at small nuclear substitution and indels and the effect of selection and population size on them. And there's a very, I'm going to jump forward here before I show you the data. The long story short is that there is no effect of population size on these small nuclear mutations. When you look at the base substitution rate or the small indel rate, again, you would expect a negative trend with population size if there was selection. We see nothing of that sort for either substitutions or indels. If we subdivide our data into synonymous changes versus non-synonymous changes, we see no effect of population size, no indication of selection. And if we include frameshift mutations, or we look at the non-synonymous changes over non-synonymous sites divided by the synonymous changes per synonymous sites, again, we see no indication of selection in any of these data sets. Finally, we also looked at the radicality of the amino acid changes. And while there might be some negative trend, again, you can't really say that there is proper negative selection on any of these kind of mutations. So as far as we could tell, no effect of selection. The same is true for the mutation spectrum. It's not really affected by population size at all. This is for the substitutions. And the only differences we could really tell, and this is across population sizes, so it's not, there's no real difference between population sizes, is that on average, the substitution rates are lower in the exons than in introns and intergenic regions. And the substitution rates are higher in the, or lower in the chromosomal cores than they are in the arms or tips. And this is, and there's no significant interaction between these, the chromosome on which the mutation occurs, which is up here, the recombination, the functional domain, exon, intron, intergenic, or the recombinational domain of the chromosome, which is illustrated in this graph. So In addition to that, we also saw a slight difference between non-germline and germline genes, in which case the germline genes on average had slightly higher base substitution rates. But there is a significant interaction between the recombinational domain and the germline genes or non-germline genes. So one thing we noticed here, though, is that the vast majority of mutations, or the highest mutation rates, substitution rates, are AT to TA mutations in this neighborhood of T and A on the 5' and 3' sides of the genomic site, respectively speaking. And most of these mutations happened right at the boundaries of homopolymeric runs, which led us to take out these mutations and reanalyze all of the data sets. And we completely lost any kind of signal of differences of mutation rate in the functional domains and in the germline versus non-germline genes. The only difference we still see, and it is now much less extreme, is that between arms, tips, and cores of the chromosome. So a large part of the mutational spectrum or the mutational differences we detect in these lines is simply due to that one type of transition, the AT to TA. Now, if we look at indel mutations, we see a very similar trend as we did in the substitutions. The exons and the cores, again, have much lower mutation rates than the other categories. And here as well, if you look at the low sequence complexity regions of the genome, again, AT homopolymeric runs, this is where most of your mutations will be located. In addition to the low complexity mutations, the other thing we noticed is that deletions vastly outnumber insertions in this data set. And this is, again, the same for all population sizes. The ratio between deletions and insertions remains approximately the same, but you do have a very strong deletion bias. And then when you look at the spatial distribution across population sizes, it also remains the same. So what we decided to do is to essentially look how the different categories of functional annotation affect the mutability of the genome. And then we looked at the mutability of the genome relative to where our mutations occurred. And when I only took the GC content, which is this bar here, I also included the sequence complexity and the repeat sequence. So essentially these three yellow ones, I could predict the mutability of the sequence by about up to 80% of the variability I could predict. If I include everything else, I go up to about 82%. So just the sequence context and the repeat sequence for these small mutations could give you the information you needed in order to make pretty good predictions of where mutations would happen. So what is the effect of NE on SNPs and indels? There is none. The mutations are mainly a result of low complexity sequence and hotspots. And there's actually one more story that we were looking at, and this is in a review. And just based on time, I'm not going to go into detail. But what is interesting is when we looked at mismatch repair deficient lines of the essentially same C. elegans, we could see that the two mutations that were generally not properly repaired by mismatch repair were AT to TA SNPs, substitutions, and, well, small single base pair deletions. So essentially the insertions were more effectively repaired by mismatch repair and any substitution other than this AT to TA. And those two mutations that are essentially not properly repaired by MMR are the ones that lead to most of the differences in genomic distribution of these mutations in our lines which do have mismatch repair. So essentially the spectrum of these small mutations seem to be a leftover of what mismatch repair did not take care of, and might be the reason, well, mismatch repair may already have purged the most deleterious mutations before we caught them in our mutation accumulation experiment, or before we failed to catch them. So just to, well, the last three very quick points just to wrap up is essentially that function size and genomic location matters in terms of how mutable a sequence is and how likely a mutation is to be retained. The effect of the mutation affects the outcome of the selection on it. That is fairly straightforward, but it's still nice to actually show it. And repair mechanisms, such as the mismatch repair mechanism, may already have adapted to take care of those mutations that may have the most deleterious effects regarding small mutations. That yeah, that pretty much sums up these three stories, and I'm just about out of time. In terms of thank yous and acknowledgements, the two profs I really can't thank enough are Vaishali Kachu, who's right here, and Overbeck Thorsen, with both of whom I worked with on these experiments. Brian isn't in this picture, or Lucille. Both of them helped propagate the lines, get the material to get the sequencing done. Brian helped with the RNA sequencing as well. And then Megan, who's right here, helped with some of the sequencing for the small scale mutations. And then, of course, as always, I have to thank Texas A&M University, the University of New Mexico, and the National Science Foundation for lab space, office space, and funding. And that pretty much covers it.
Thank you. Thank you, Anke, very much for your very complete presentation. It's a lot of interesting research, and there's several lines of research that will follow in the future, in which you are already also working on. We now have time for some questions, if there are some in the YouTube channel. I invite you once again, those that are following our presentation, that you can ask your questions on the right here on the YouTube chat, the window that you have on your right. We have one question for now, and I will now ask the question and invite Anke to answer. It's a question by Yves Shell. So if the MA had been done under oxidative stress, do you think we would expect to see slopes more negative on non-synonymous mutations or population size graphs? Thank you.
Well, I would imagine that's a good question. So for the mitochondrial mutations, I wonder if so for the mitochondrial mutations, I wonder if that would lead to lower tolerance for some of the mutations that we're seeing, which may actually flatten the graph, because the size 1 might even be weeded out more effectively, given that you're working with a population of mitochondria rather than one single genome as you are in the nuclear graph. For the nuclear mutations, I am not sure, to be honest. I guess it depends on where the mutations are located. Yeah, I'm not 100% sure, to be honest. Yeah.
Thank you, Anke. And it's a sign. Yeah. Sorry, I interrupted you.
Oh no, I said I might just be overthinking it right now. But yeah.
But it's an interesting question, and it's a sign that it's something more to explore. So there are always new things to explore. And in the meantime, I invite those that are watching, if you want to leave questions here on the YouTube chat. For now, we don't have more questions, but I would like to thank again you, Anke, for your presentation. I don't know if Ivo wants to add any other comment before we close the session, if we don't have more questions. So if Ivo doesn't have any more comments, I would like to thank again Anke for your presentation. And we are looking forward to see the new results. You mentioned some papers that are now under review. So we are looking forward to follow the next papers. Thank you, Anke. Thank you, Ivo, for also presenting Anke and being with us. Thank you, Mariana, who is on the backstage here helping us with the connection. And thank you all of you who watched and followed this presentation. And stay tuned for the following events following Ciência meetings online.